And we are now recording, and thank you very much, Hagan, for agreeing to give a Build a Cell seminar, and it's all yours.
Okay, so this is Hagan Bayley talking to you from Oxford, where it's 90 degrees outside and probably about 110 degrees in my office. So if I fade away halfway through, you'll know what's happened. So today I'm going to tell you about designer tissues, how we can make synthetic tissues, and how we can make modified living tissues and perhaps make hybrid tissues between those two things. And whoops, now my slides are not moving. Hold on, there we go. And Kate asked me to introduce myself. So let me just tell you that we're a lab of about 20 people and maybe about half graduate students and half postdocs. And our underlying interest is in membrane proteins. And so from the basic science point of view, we're interested in how membrane proteins assemble and function and their structure, but we're also very much interested in membrane protein engineering. And from that, we've developed a lot of biotechnology. And so we've been interested in using membrane proteins for sensing and DNA sequencing, and more recently for looking at single molecule chemistry, bond making, and breaking reactions. And we've also, as you'll see in this talk, been using membrane proteins for making designer tissues, and we're also interested in developing high throughput screening techniques for membrane proteins. But the main thing that holds us together is working on membrane proteins and applying chemical, biological, and physical techniques. So my slides don't seem to want to move. Let's try that. Very strange. Let me try the little arrow at the bottom. There we go. Okay, so designer tissues. Well, there are three areas that I'm going to talk about. So the first are purely synthetic tissues that are made from parts. So these will be non-replicating materials, so they might be safer than biological tissues. And we like to think that the materials that we're making are biocompatible, they're pretty robust, and they have functional properties which are emergent properties. So they're properties that you won't get from individual compartments. Using some of the technologies that we've developed for synthetic tissues, we've also been printing living cells, and we believe that we can do that with high viability and at high density, and we can pattern these living cells in 3 dimensions, as you'll see later. And one thing that we'd very much like to do is bring these two things together to make hybrid tissues, to integrate our synthetic tissues with the printed cells. So first of all, let me tell you about the synthetic tissue area. So there's been a lot of work on synthetic cells, which many of you are involved in, but there's been much less work, I would say, on synthetic tissues. So we might ask, what is a tissue compared to a cell? Well, it's a collection of compartments rather than individual compartments. And these compartments can interact with each other and the environment. And that allows these materials to have emergent properties which are greater than the sum of the individual parts. And there are two ways that I think of looking at synthetic tissues. On the left, we have separated compartments or cells in a matrix. And another type of tissue is comprised of closely packed compartments or cells. And they're shown on the right-hand side. And that's what we've focused on. So another issue are the constraints in the synthetic cell and tissue field. And I think different people have different rules for their building blocks, which depends on their goals. So some people would like to mimic and understand how present-day natural cells work. Other people would like to understand early cells and the origin of life. And other people like us really just want to make interesting and useful tissue-like materials and devices. And so we can ask whether we have to rely on biologically derived components and natural assembly rules, or can we use unnatural components and new different assembly ideas? And I would just say in our lab, just reiterate, we're just interested in making interesting tissues, and we don't put any constraints on what we can use to build our cells and ultimately our tissues. So we can use any unnatural materials and put them together in any way that that we want to achieve our goals. So let me first then talk about the synthetic tissues. And these in our lab are made from 3D printed droplet networks, and I'll tell you how we got to that position. So these are aqueous droplets, and these droplets are connected together by individual lipid bilayers. So they're not connected by double bilayers, as would be the case if you brought two cells together. They're connected by individual bilayers. And then we can add to these functional components. So we can encapsulate enzymes, we can add protein pores, transporters, and so on. And the pores, as you'll see in some of the examples I show you, will allow these droplets to communicate with each other and indeed with the outside world. So this was derived from a very simple idea, and we got the idea for that we could add individual lipid droplets. These will be of the nanoliter to picoliter volumes to an oil containing an amphipathic molecule. And the idea was that when you add the droplet to the oil, the amphipathic molecule will form a monolayer around the droplet. Obviously, the hydrophilic head groups will be in, the fatty acid tails will point into the oil. And a typical amphipathic molecule that we might use and will be used throughout this talk would be a phosphatidylcholine. And then when you bring two of these droplets together, they form a bilayer at their interface. That seems obvious at first sight, but when you start thinking about the energetics, it's not immediately obvious that that would occur. But they do indeed form a tight bilayer between the two droplets, and that's the basis of our synthetic tissues. So my slides have stopped moving again. Let me try once more over here. Here we go. So when we first started this, we started making some very simple structures. I'll just show you one example here. So this is a 4-droplet system, which will rectify an electrical potential. And to do that, we incorporate into the bilayers between the droplets an engineered alpha-hemolysin pore that we call our 7R diode. Now, hemolysin is a membrane pore that contains 7 subunits. Each of these subunits that we've engineered to contain 7 arginine residues projecting into the beta barrel that goes through the membrane. So there are actually 49 additional charges in this pore. And it was an example of chance favoring the prepared, because we were trying to use this molecule to slow down DNA for DNA sequencing. That did not work very well. But we discovered that this pore would only conduct current when you apply a positive potential to the bottom side, to the trans side. When you apply a negative potential, it just closes up, so it acts like a diode. So when we assembled our droplets in this manner, and I can tell you that this pore will insert vectorially into membranes because it has a big extramembranous domain. So it's possible to assemble these systems with the pores pointed in the direction you want to point them in. You end up with a simple system that will rectify a current. So if you make this system from parts from Radio Shack and apply a sawtooth voltage, it will rectify that in terms of the output current. And if you make it from droplets, it will do the same thing. So this, we like to think is an example of a simple device built from droplets that has emergent properties. So it will do more than the individual droplets can do. And indeed, it's made from very simple parts, just oil, salty water, some lipid, and a very simple engineered pore. So we wanted to go on from that to make more complex devices. And to do that, students at the lab called 𝗁𝗶𝗀𝗀𝗀 devised a 3D printer to print picoliter volume droplets. So typical size that we would use would be about 50 picoliters. They're about 45 microns across, these droplets. And again, we're printing into oil, as you can see here. And this device gives us the possibility of printing many thousands of droplets, and it also gives us the possibility of patterning these droplets by using multiple printing heads. So here are some examples of what you can do here. Here's the general idea on the left. The droplets are ejected into the oil, a monolayer forms around them. When this droplet hits the droplets that are already printed, it will form bilayers which will hold it into place and also give you the possibility of allowing these droplets to communicate through channels and pores. And here's an example of what looks like a arborizing part of the nervous system or vascular system. And this is printed in 20 layers, which you can see here from our printer. And here's another example of just a three-dimensional shape that looks different in the X, Y, and Z directions. And you can see the result of the printing on the right-hand side here. You can, ultimately, of course, we'd like these systems not to be in oil. We'd like to be able to use them for physiological or medical applications. So we'd like to get them into water. It is possible to print inside an oil drop that's in an aqueous environment. And it is also possible, and we're working on improving this in transferring these printed structures to an aqueous environment. But I just wanted to point out here that once these 3D printed structures have been transferred from oil to water, the monolayers that are on the outside will become lipid bilayers because they will take up more lipid molecules so that the hydrophilic head groups can point into the aqueous phase. So once you've transferred one of these structures to water, there are bilayers between each droplet, and that allows you to put in protein pores to get them to communicate, but there are also bilayers facing the outside world, the aqueous world, and again, you can get them to communicate with the outside world in that way. Now, oops, there we go. I should say that a lot of this is easier said than done, and we're still working on improving these techniques. And the fidelity of this printing is affected by the oil that you use, the lipid, the contents of the droplets, with the salt they contain, the proteins inside them, the temperature, whether you apply an electrical potential or a physical pressure. And just to show you one example that we published recently to try to improve this printing, is if you examine droplet pairs, depending on the lipids that you use or the oil that you use, there will be a different contact angle between these droplet pairs. So these droplet pairs in oil is a lipid bilayer between the two droplets. If the contact angle is very small, when you print in 3 dimensions, you get quite disordered structures, maybe not surprisingly, because these are not packing very well. When the contact angle is very large, again, you get disordered structures because of the droplets pulling, each droplet pulling on its neighboring droplet. And it turns out that there is a sort of sweet spot, a contact angle that's about 35 degrees that allows you to print with much greater fidelity. Another thing that we found out during this study is that if you have defects in the first layer that you print, they will propagate through subsequent layers, and that's another thing that really affects the fidelity of the printing. And also from this paper that's just came out is this beautiful image that shows what a fragment of a 3D printed material looks like. So these are droplets that have been printed and then photopolymerized, and this is a fragment of them. And you can see, and of course, if you think about it, it's obvious that these droplets are not just like squashed soccer balls. They are actually a dodecahedra when they're packed very tightly. And you can see that beautifully in this image. You can also print droplets on a millimeter scale and then build them into larger structures by hierarchical assembly. And this is something that is just being completed in the lab now. But just to show you some early examples, if you print these 2 structures on the top and bring them together, they will fuse with each other, and they will anneal at this interface. And then you can make larger structures that we'll come back to later. If you make a chain of these, you can make long structures from them where the interfaces are all annealed, so there are actual droplets at the interface rather than broken droplets. Of course, these can serve as modules to make different shapes. And also, interestingly, if you take one of these structures and cut it and bring it back together, it will self-heal, so it will re-anneal at the interface. And this will allow us to make centimeter scale structures. Now, we'd like to give these structures interesting properties. And one of the first things that we looked at was to give them mechanical properties. And this is based on the idea of a lot of these people working in soft robotics. And you can see an example from this Nature review here. This is, of course, nothing like printed droplets. This is a much larger creature that they've made that will swim around. But we'd like to be able to make droplets that can change shape and grip things, and just like these soft robotic devices. And so here's an early example here where we printed droplets, some containing high salt, some containing low salt. And after they're printed, water will move from the blue droplets to the orange droplets. And in the top case, this will fold like a bimetallic strip. It's a roughly 2-dimensional structure to form this circle on the right. And more interestingly, this flower-like shape that's printed onto a non-sticky surface on the bottom will fold up into a hollow sphere. And there are really 2 purposes for doing this. One is to make shapes that you couldn't possibly print. It'd be very hard to print a hollow sphere, but also ultimately to print materials that mimic muscle. Now, those shapes, those devices change shape very slowly. And what we've been working on more recently is to use so-called smart polymers like polynipam, poly-N-isopropyl to get printed devices to change shape. So this polymer, when you heat it, will shrink, as shown on the top is the droplet pair that's printed with the precursor, then photopolymerized, and then you heat it up and the droplets shrink. So you can obviously print droplets with and without the polymer, so only one of these two droplets will shrink. And you can also print materials that have different formulations of polynipam in them. And then when you heat them up, as in this case, this one will actually curl up. You can do that with heat, or you can do it with light. So you can use gold nanoparticles that will turn light into heat. And here's an example on the top right here, where again, we can get this object to curl up when we shine light onto it. And this is done because the gold here is actually absorbing light and then heating up this structure. So having made these things, we can get them to perform interesting tricks. So here's one here that, again, contains two different polynipam formulations and also a magnetic droplet as well. And we can get this device to shrink, to go through a gap, to curl up, to shrink again and come out the other side. And then we can do more elaborate things. So here we got a little maze. This structure is guided through the maze by a magnet, but it can shrink to get through the entrance. It can go around the maze. It can pick up an object and then take it to another site, drop off the cargo, and then eventually emerge from the exit to the maze. So we'd like to be able to eventually, obviously, to use these in medicine to deliver particles to certain sites in the cell, but obviously that's quite far off at this point. Another thing that we can do with these materials is to send signals through them. So if we print some droplets with our 𝗵𝗆𝘁𝗵𝗲𝗁 and some without, the green ones with, in this case, we can put two electrodes on our piece of synthetic material, apply a potential, and this will conduct an ionic current through the green pathway. So it conducts a signal very rapidly and directionally. And of course, this is what a nerve or an individual neuron will do by a completely different mechanism, but this serves the same purpose. And to go back to these assembled constructs before, we can also now assemble long pieces of material that would contain a conducting pathway through the middle, so we can conduct currents across larger distances, centimeter distances. Now, this has also been done by using purely hydrogel structures rather than printed droplets. So these devices have droplets at the end of these so-called artificial nerves. And in this case, instead of applying a potential, we're using a bacteria rhodopsin-like protein to pump protons into this structure. So bacteriorhodopsin, or this AR3 protein, is a light-driven proton pump. So it's an electrogenic proton pump. So when you shine light on it, it moves charge into the structure. And if you connect this with an external circuit, a current will be produced that travels through the structure. And you can also get these structures to release transmitters at the end. I won't talk about that today. But another interesting thing you can do is pack several of them into an elastomeric sort of elastomeric nerve, And that allows you, if you can address each one with a different LED, to send different signals through the same pathway. And here in the bottom right, you can see 3 different types of pulses being transmitted simultaneously through the same artificial nerve. So based on those ideas, we would like to kind of expand this idea of making synthetic tissues where we can have various inputs, transmit and process signals within the tissues, and then have various outputs. And we would use these, for example, to transmit electrical signals to living cells, to produce muscle-like activity, or maybe induce these materials to make and release drug molecules in a patterned manner. So I think a couple of weeks ago, you heard from Mike Booth, and one way that we've been looking at this is to use his light-activated promoters, and that allows us to put a transcription and translation system into these droplets, and we can do that in a patterned manner. And as he will have shown you, when you shine light on these structures, you can express, in this case, a yellow fluorescent protein in a defined part of the structure. And that allowed Mike to also express the 𝗁𝗆𝘁𝗁𝗂𝗃 pore, which is of course colorless, again in a defined part of these 3D printed structures. And that in turn allows you to transmit an electrical signal along this pathway. And so this is an example. It's irreversible in this case, but it's an example of light-activated signaling. So I thought, because you'd heard about that, and this doesn't want to move again, let's try once more. I'd tell you about another signaling process that we're working on at the moment that really stemmed from the covalent chemistry work in the lab. So the idea here is that we have our protein pore in the membrane. We put this molecule, which I'll tell you what it is in a moment, into the pore and hold it at the top, and then cut it specifically so this part of the molecule goes across the membrane and transmits a signal. Now, we started off trying to cut phosphodiester, amide, and ester bonds. That turns out to be very difficult to do, but we ended up in the end doing this with disulfide bonds. So the idea of the chemistry is to have a disulfide, cleave it with a thiol. The thiol will be on the interior of the protein. It will be a cysteine residue, and then it can cleave either by attacking this sulfur or this sulfur to produce 2 different possible products that you'll see in a moment. So if we can put this inside our nanopore tube, our aim at the time was to get site-selective cleavage, so it would cleave this disulfide bond and not this one, but also to get regioselectivity, so it would attack this sulfur and not this sulfur. So what we're doing here is making this large molecule. So it has a peptide segment in the end, of which the length can be adjusted, and a biotin that will bind a monovalent streptavidin. And then here's our disulfide that we're going to cleave. And then in this particular molecule, there's a DNA and oligonucleotide of 4-mer at this end. So the idea is the protein goes into the pore. There's sulfhydryl inside the pore. It will cleave this bond if it's aligned correctly. If it's not aligned correctly, it won't cleave it. And it can cleave it either to release the bottom bit or release the top bit. If it releases the bottom bit, that will produce a signal on this side of the membrane. And to get a long story short, this was achieved in the lab, and you could make both site-selective and regioselective cleavages. And I won't go through all the details, but if we take one of our constructs with a cysteine at residue 121, it will cleave completely regioselectively at this sulfur atom. But if we take the same construct and put our cysteine at position 113, it will attack this top sulfur atom rather than the bottom one. So the idea is in our droplet system is to have pores that contain cysteines that will selectively cleave these molecules and release, in this case, a piece of DNA into the trans side that will bind to a defective promoter and then turn on transcription and translation and produce some sort of interesting product. So we're in the middle of doing this now. We'd like to produce orthogonal input signals because we can move our cysteine within the pore. We need a strong signal to turn on transcription, for example. So we have to have multiple pores here, which is proving to be a bit of a problem. And then there are different outputs. We can do promoter complementation, but we can also do protein complementation, and I think we can also transmit entire ribozymes or enzymes across this structure. Okay, so that's what I'm going to tell you today, at least about synthetic tissues, some old stuff and some things that are still in progress. Let me now tell you about printed cells. So basically, we've been using the same 3D printer, but it's been quite highly modified in order to print cells. And the cells are usually printed in quite viscous extracellular matrix, which means that you have to have a much more powerful piezo on your printer. And also, a lot of these hydrogels are very temperature dependent. They will either gelate at high or low temperatures, depending what they are. So you need a very good temperature control over this printing. But to cut a long story short, we can print viscous materials. We can vary the droplet size down to below 10 picoliters, and we can print cells in these droplets that are very high densities, up to 40 million cells per mil. And once they're printed, we can say we're using matrigel. We can print at low temperature, then bring the temperature up to gelate the matrigel, the extracellular matrix, and then we can replace the oil with an aqueous medium so that the cells will now grow in 3D culture. So on the left side here, you can see that the lipid that was used to encapsulate the droplets originally is just using a red dye to show the lipid bilayer. And here you can actually see the individual cells inside the droplets. Once they're transferred to medium, the lipids disappear. They either dissociate or they get eaten up by the cells. And now we have a nice 3D culture of our cells that we're interested in. So here is an example that was just published very recently of some human neural stem cells that are printed in that way. So you can see at day 4, you can see these individual cells after printing, but as time goes on, day 8, day 14, these cells start putting out processes and looking very much more like neurons. And you can see the length of the processes increases greatly with time. This is at day 14, where they have an average length of about 200 microns. And these cells, once they're printed, are functional, and they will show spontaneous calcium oscillations using the dye, the calcium-sensitive dye, 𝗶𝗏4. When they're first printed, you see few of these oscillations, and they're not very highly correlated. But once they've been in culture for some time, this is after 44 days in culture, you see many more of the calcium oscillations, and they become highly correlated, as you can see in these individual ROIs, but you can map this by using a computer program and see that these cells are really signaling to each other in the three-dimensional cultures. The cells will migrate. So if you print the cells surrounded just by extracellular matrix, the cells will first put projections into the matrix, and then after about a month, they will start actually migrating along these projections into the matrix and fill this outer layer that was originally just ECM. And if you print astrocytes in the outer layer, shown on the bottom here, the cells will project and migrate, but they will also bundle in the cultures to form structures that very much look like bundled neurons inside nerves. We can print these in different patterns to test ideas about how these structures develop. So we can print patterns where the astrocytes on the outside are on the outside. In this case, the astrocytes will stay put. The cells will send projections into the astrocytes and eventually migrate. But we can print this in the reverse pattern with the astrocytes on the inside. And in that case, the projections will move into the astrocyte layer. The astrocytes will stay put. And then eventually these cells will migrate inwards in this case, and outwards in this case. So in our sort of printed cell projects, right now we're very much focused on printing neurons and trying to, for example, to imitate the layers of the cerebral cortex and so on. So there should be more of that you can hear about in a year or so's time. So let me just finally finish up by telling you about this idea of making hybrid tissues. So we can make synthetic tissues, and we can print cells. Can we bring these together in some way? So the idea would be to connect synthetic tissues to printed cells, or connect synthetic tissues to living tissues, and then maybe be able to control them from the outside world, as I mentioned earlier, and use these to repair or enhance damaged organs. So one way that we're looking at, there are several different ways that you might do this, what we have been looking at is to be able to mimic the gap junctions that will span certain cell junctions in the body. So if you, cells can be electrically connected by gap junctions, and here we see two cells. There's the outer membrane, plasma membrane from one cell, plasma membrane from another cell, and gap junctions are proteins that will span these two so that you can transmit an electrical signal directly from one cell through to another cell. And we've been trying to mimic that, again, by using engineered pores. And one way that we have looked at, and I think is we should be pursuing some more, is to take our hemolysin pore, to mutate it. And the original idea was to put a cysteine on each subunit, each of the 7 subunits, and then connect the pores with, say, nickel and NTA, or 2 oligonucleotides, or and so on. But again, we were lucky with this, and when we expressed this protein in E. coli, it actually came out as a 14-mer that meant that 2 of these 7-mers had got together in some way. And again, if you think about the geometry, there are different ways in which these can get together by forming disulfide bonds. And if there are just 1 or 2 disulfide bonds in these structures, they can form very sort of loose hinged arrangements. But if you have 3 or 4 or more disulfide bonds, the only way in which you can arrange these subunits is with their central axes aligned. And when we analyze these, they had an average of about 4 disulfide bonds. So the redox potential in the cell was just right, really, to allow these bonds to exchange and exchange and exchange until they form these dimeric structures. So they're dimers of heptamers, so they're 14-mers. And I would remind you that I told you earlier that if you take these synthetic tissues or other tissues in normal tissues in water, they will have a bilayer on the outside. And so the idea has been to take these dimers. So here's a class average of side-on EM views of these dimers of heptamers, and so there'll be a lipid spanning domain here and a lipid spanning domain here. And here's a top view of the structure. You can see the 7 subunits very nicely here. And then if you take liposomes, you can get these structures to connect, on a good day at least, to connect these liposomes to each other. So the idea eventually would be to take these printed cells and the synthetic tissues and connect them to each other electrically through these kind of structures. Okay, and then build these hybrid tissues in that way. So let me finish at that point. So I've tried to tell you that we can make both synthetic tissues and cellular structures by 3D printing at high resolution. We have been getting some way towards getting these hybrid structures and cellular structures to communicate with each other. And one of our main goals at the moment is to apply these materials in neurobiology to release drugs or repair damaged parts of the brain. And so I'll just end by saying thank you. Here's another view of the group, and thank our sponsors and thank everyone who's worked on this, and say that some of the work was a collaboration with Mike Booth's lab, who spoke with you a couple of weeks ago, and with Francis Zeller in physiology and anatomy and genetics here, and Kevin Foster. I didn't mention that work. Actually, we can also print bacteria in the biochemistry department. So thanks very much. I'm going to try and escape from here and take any questions.
Thank you very much, Hagan. That was amazing. I have a few questions, but you also have already a couple questions in the chat. Can you see the chat window?
I'm trying. This computer is
I can read it.
Things in different places to my last computer. I've got participants, security. I've got everything except chat.
I'm happy to read it.
Okay.
So question from Joe. On synthetic tissues, you mentioned unnatural components. Have you tried more interesting emulsifiers outside of phospholipids, for example, nanoparticles, polymers, etc., or making all aqueous printed tissues using aqueous two-phase systems?
Yeah, I'm not sure I took all of that in. It's a little bit hard to hear you, but basically, no. We started off actually just by using some very simple amphipathic molecules. Then we switched to lipids, which are more like natural bilayers. But I think we could probably make much more stable structures using block copolymers, for example. And indeed, that's what Oxford Nanopore is now using on its sequencing chip, it's using block copolymers instead of lipid bilayers. So I think there are many different possibilities. We have put in different lipids, so we can make asymmetric bilayers. We can make bilayers containing cholesterol and so on. You'll realize that if you bring two droplets together that have different monolayers, it's a very nice way of making an asymmetric bilayer, and all bilayers that we know of in nature are asymmetric with respect to the lipids, and it does affect protein function. So we've been interested in that.
Thank you very much. Another question, can you hear me?
Yes.
I switched my microphone, that should be slightly better.
You're a bit quiet.
I can speak up a bit. Okay, so next question. Have you tried encapsulating smaller liposomes inside the droplets to mimic organelles? Also, is there a way to induce a water-filled gap and control the thickness of the gap between droplet bilayers transferred from an oil to aqueous phase?
Okay, the first part, yes, you can put lipid vesicles inside the droplets. We've been having some problem with them fusing with the bilayers, but you can get around that by using, say, pegylated lipids or a good choice of the phospholipids that you use. The second question was, can we make an aqueous gap? But these droplets are connected by a single bilayer, so you can't make a gap, but we certainly have in the past been interested in making systems that have two bilayers. One reason is to mimic natural structures like the nuclear membrane, which has pores that span two bilayers. But another item of interest would be to sequence the same piece of DNA twice by having it pass through two bilayers. You need an electrode between the two bilayers to be able to capture the signal, I think. So we're not working on that now, but we have been interested in that area. Yeah.
Okay, next question. Are you using these structures for biosensing applications?
Not really. I mean, we've been very interested in the past in using nanopores for sensing. And in fact, when we first formed Oxford Nanopore, the company was really formed to sense small molecules. And you can sense metal ions, organic molecules, little bits of DNA, peptides, or whatever. And it was only after about a year that we decided to really try and sequence DNA and RNA. And the company's kind of turning full circle now, coming back to sensing other molecules. It's a general sensing platform, so looking at sensing small molecules and looking at polypeptides as well as DNA and RNA. In terms of sensing with these structures, really people do ask whether you could use these structures to sense things, whether you can use them to compute things, and whether you could use them as factories to make drugs and things. And I tend to think that's rather impractical. But if you look at the entities themselves, you would certainly like them to be able to sense things for their own purposes. So if they could make drug molecules, for example, you'd like them to make those molecules in response to an outside signal. So we'd like to print synthetic materials that can detect signals from the outside world and process them in some way. So think about them in some very simplistic way. And then as a result of that processing, decide whether they're going to synthesize some growth factor or whatever and release it into the tissue that they're located in. So we're not thinking of using these as sensors or as factories, but we'd like them to build that capacity in them for their own purposes.
And another question. A great talk. These droplets look very robust. Could you share the composition of any of the aqueous droplets you used? Thanks.
Well, for most of them, we've been using 𝗵𝗃𝗀𝗃𝗀𝗃choline, the same lipid that we use for bilayer recordings. But as I said, you can use many different amphipathic molecules. Their stability does really vary depending on the contents, and I don't think we've completely made that foolproof. So it may be that there's some block copolymer formulation or some head group polymerizing process or something that will make these structures much, much more stable. So I would say they're pretty stable, maybe much more stable than you would think. But I think if you wanted these, for example, to sit in a human body for 2 years, that's a big ask at the moment. I think you'd really have to improve the stability to get that to happen. But I think if you want something that will last for a few days, yes, that we can do.
Thank you. And another question. In the hybrid tissues, do you anticipate an exchange of the membrane material between the real and synthetic structures, or would the gap junction mitigate this?
Well, in principle, given the structure on paper, the lipids should not exchange. And, but yeah, it's definitely it's a possibility that when these two bilayers are brought so close to each other, there would be some limited exchange between them. I mean, phospholipids dissociate from bilayers incredibly slowly. If they did dissociate in a gap junction, they wouldn't have much, they wouldn't have anywhere to go except back into the same bilayer or into the opposing bilayer. So there might be some localized exchange, but I would expect it to be slow.
Okay, next question. What are the biggest inefficiencies in assembling the synthetic and hybrid tissues? How might these be overcome in the future?
Well, I think right now it's just that's been at a lab stage, and we haven't really worked on that for a couple of years. I think it's not an efficient process at the moment, and I think if you wanted that to work well, yeah, you would have to improve the insertion of these pre-assembled pores, and there might well be ways to do that. So there might be some way of having some ligand on the recipient membrane, and so you can bring the two bilayers very close together and then increase the efficiency of the insertion of the membrane spanning protein. But that's something that definitely requires more work to make that more efficient.
So this is all questions in the chat. I would like to take advantage of being unmuted and ask you my own question. I really like this light activated patterning that you were doing. What's your feature size on that?
Well, Mike, when he was in the lab, showed that you can just do that in 2 dimensions at the level of individual droplets. So what we'd really like to do now is to be able to replicate that in 3 dimensions, and that's less easily done. So there are really 2 ways to do that. One is you can kind of print the feature you want to light up. So when you shine light, only that droplet will light up. And then if you want to do it some expensive way, you could use some very fancy microscope, but we don't want to do that. So the other way to do it is maybe to use orthogonal light beams of two different colors. So to get a droplet to do something might require, 300 plus 500 to deprotect some compound or so, perhaps. So, and then I think you can get limited success in 3 dimensions just by using a scanning focus laser beam as well. So the answer to the question is, in 2 dimensions, yes, you can get very high resolution, but in 3 dimensions, we need to do more work.
Thank you. A couple more questions in the chat. To what degree of certainty would you say that artificial cells or tissues will be used as implantable materials in the foreseeable future?
Oh, gosh. That's what reminds me of that adage that the hardest thing to predict is the future. Yeah, I think, gosh, when you look back on what's just happened in science over the last 20 years, 50 years, it's absolutely incredible, So, I mean, if you look at, even look at DNA sequencing, used to cost 3 billion, take so long to do. So I think it's very likely that we will be able to print cells that have been altered, using genome editing and make all sorts of printed tissues, some living cells that can be put into the human body or into animals. And then I think these sort of more bottom-up synthetic tissues will become much more stable and much more sophisticated, and we'll be able to maybe address them in ways that you can't, where it's hard to address living tissues. And I also like the idea of these hybrid materials, where you don't worry about whether you're looking at the origin of life or something. You just make some material that can do something really interesting. So I think it's quite possible that, for example, someone could have some messy brain tumor removed, and then you could implant some synthetic tissue and that will make drug molecules, and you could control that from the outside world using magnetism or some other physical, or you can control them with chemicals too. You can just give someone a pill that will, this idea of chemogenetics, where you can trigger off molecules using orthogonal drugs and so on. So yeah, I really do think, I can't tell you when, but I would be surprised if within 10 years we weren't using these kind of materials.
Thank you. Another question in chat. Could you somehow use a constructive and destructive interface of light to make those 3-dimensional light-induced structures? Is that the fancy microscope approach?
I think I see what you're saying. Well, the fancy microscope approach, I'm just thinking of using, some multiphoton microscope or some confocal microscope, So, but you really want something you can implant in someone and just really, you or they can control that from the outside world.
Another question in chat. Are there particular disorders or diseases that the lab is particularly interested in developing the research towards, or is the lab looking very broadly to explore what works best?
Yeah, I think in our lab we do tend, I hope, work very broadly, just work on new ideas and so on. And then I think if something is useful, which is rare, but there are lots of scientists doing interesting things, then's the time to commercialize it. And that certainly was the case with the DNA sequencing. You can do some interesting things in the lab, but to commercialize it, to take it to the state where you've got a machine that you can sell that's foolproof and the error rate is reasonable, definitely requires spin out company or so on. So I think really on the whole, our goal has been to do new things in the lab, which I think is what academics should be doing. And of course, we're limited by really the amount of the funding that we have, most of us are at least. And then if something interesting comes up, then I think it's a good idea to try and form a company where you can, put 10 or 100 times more money into the project.
Thank you very much. It looks like there are no more questions in chat, and we're up to the hour right now. So I want to thank you, Hagan, again, very, very much for giving this awesome seminar. This was really inspiring, and I hope to see some of that really applied to making complex synthetic cells, maybe one day self-reproducing.
All right, thanks, Kate.
Thank you, and stay safe, everyone, and have a good week.
All right, see you all soon. Bye now.
Thanks, bye.